Assalamualaikum and hi everyone, I am Rosin Rosli from S3T11 and this video is about how DNA replicate. DNA has 4 nucleobases, adenine, thymine, guanine and cytosine. The nucleobases in a DNA strand have preferred partners to form hydrogen bonds with. Adenine pairs with thymine and cytosine pairs with guanine. These are the base pairing rules that allow DNA replication and protein synthesis to happen. According to the semi-conservative replication model, the two original DNA strands separate during replication. Each strand then serves as a template for a new DNA strand, which means that each newly synthesized double helix is a combination of one original and one new DNA strand. Mechanism of DNA replication have 4 steps. The first one is replication fork formation, then primer binding, elongation, and termination. This is an enzyme called helicase is to separate the two strands and making them available as template strands. This enzyme is catalyzed in relieving this strand by breaking, swiveling, and rejoining DNA strands. This enzyme is known as topoisomerase. The process begins when this enzyme makes a small piece of RNA called a primer. The primer is the starting point for the construction of the new strand of DNA. This enzyme is known as primase. This enzyme binds to the primer by adding DNA bases and make the new strand of DNA in one direction from the 5' end to the 3' end. This enzyme is known as DNA polymerase III.
Hold up, wait a minute.
Then what happened to the other strand? The strand which is start from 3' end to 5' end is called a lagging strand. It is the opposite direction from the other one. This strand cannot be made in this continuous way, and it is start with an RNA primer, so the DNA polymerase can only make this strand in a series of small lumps known as Okazaki fragments. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction, and the process is repeated again. Once the new DNA has been made, this enzyme removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. This enzyme is known as DNA polymerase I. Finally, this enzyme seals up the fragments of DNA in both strands to form a continuous double strand. This enzyme is known as ligase. And the new semi-conservative DNA are formed. Here is the differences and similarity of leading and lagging strand. On leading strand, it is synthesized continuously towards replication fork, while on lagging strand, it is synthesized discontinuously away from replication fork, and no formation of Okazaki fragments on leading strand, while it is several Okazaki fragments are formed on lagging strand. Both are require RNA primer to initiate replication. Here is the function of enzymes involved. The first enzyme is helicase. Helicase is catalyzed the untwist of the double helix of the parental strands, and it is separate the two parental strands and making them available as template strands. Second is single-strand binding protein. It is hold unzipped single-stranded sections of DNA apart during replication. Third is topoisomerase. It is stabilized DNA helix as it is unwound by helicase enzyme. Next enzyme is called primase. Primase is synthesis of short RNA primer by use the parental strand as a template. It is generally 5 to 10 nucleotides long. Next is DNA polymerase III. It synthesizes new DNA strand by adding nucleotides to an RNA primer by using parental DNA strands as a template. Last but not least, it is DNA polymerase I. It is remove the primer and replacing it with DNA nucleotides one by one from the 5' end to the 3' end. Lastly, DNA ligase. On leading strand, it is catalyzed the joining of 3' end of DNA that replaces primer to rest of leading strand DNA. And on lagging strand, it is catalyzed the joining of Okazaki fragments.